Good afternoon, ladies and gentlemen. I'm glad to have the opportunity to share our experience about Ebola vaccination, more specifically about Zabdino and Vabia, which is the vaccine regimen that we have developed in Janssen. My name is Valérie Auriol-Mathieu. I am a medical doctor and I am Global Medical Affairs Leader for this vaccine. I work full-time for Janssen. The first part of my presentation will be about Ebola viruses and the disease itself. For the second part of my presentation, I will speak about the vaccine. So first of all, let's start with the virus. And like Marburg virus, Ebola viruses belong to a big family, which is Filoviridae. The genus Ebola virus was named after a river which is passing by the place where Ebola was discovered for the first time in 1976. So it's a recent history for a human disease. Since this time, there has been 6 species that have been identified. First of all, 5 species in human: Zaire, Sudan, Typhorus, Bundibugyo, and Bombali. But also a species which is infecting only non-human primates, Reston. And what is important to remember is that the largest ever Ebola epidemic in West Africa from 2013 to 2016 was caused by Zaire Ebola virus. There are some variants of Zaire Ebola virus, Kikwit, Mayinga, Makona. Nevertheless, they all belong to the same species. Here it's an illustration of what I just explained about these different species in the family of Filoviridae. Nevertheless, the diseases that are caused by these different viruses are very, very similar clinically. So Zaire Ebola virus and Sudan Ebola virus, for example, they were both identified 50 years ago or so. And Zaire is really the most frequent, and then Sudan is the second frequent. And in terms of clinical presentation and case fatality rate, they are very, very similar. Marburg was identified even earlier in Germany, close to the Marburg city, and it caused also an outbreak in Angola with a very high fatality rate, apparently even higher than with the Ebola virus family. But there is very little experience with Marburg virus, fortunately for all of us. This is illustrating the frequency of Zaire Ebola virus. And here, Zaire Ebola virus is in pink. So you see that in West Africa and also Central Africa, it was really most common. And the size of each bubble is representing the size of the epidemic. So the second thing that we can see in this map is that most of the epidemic were always small in the history. Even the epidemics with more than 100 cases were relatively infrequent. How does the transmission of Ebola virus work? So first of all, a lot of research has been done to try to identify the natural host, the reservoir. And the suspect is the fruit bats. It's really the most likely natural host. How does the transmission between a bat and human can happen? We think that it can happen either directly through a bat or through another animal that has been infected by a bat and that is then hunted, for example, by human people. So the Ebola virus is only transmitted through body fluids and secretion containing the virus. So for example, it's blood, it's feces, it's vomit, and it's really the major sources of contamination. It's not transmissible through aerial transmission, fortunately. When people die from Ebola, their dead bodies are highly infectious and are often the reason why an Ebola outbreak is spreading because of some burial rituals. And people can transmit Ebola for some time, even if they recover. And some people are even long-term transmitters of Ebola because they have persisting infection, even if clinically they have recovered, and it can last, for example, for very long in the eyes or in the semen. So the people who have recovered from Ebola should follow some regular clinical assessment to really monitor if they are still susceptible to transmit the disease. So breastfeeding is contraindicated. And because of transmission in semen, people should use a condom until they are not carrying Ebola anymore. This is just a graphical illustration of the virus ecology, showing that from the natural host, which is the bat, we think that animal-to-animal transmission can happen, for example, to non-human primates or to apes, and also to other animals like bugs that are hunted for food. Human can also have direct contact with bats. And then from a first person who is infected, it's easy to be infected by Ebola with a human-to-human transmission for the people who are taking close care of the sick people. So, for example, the healthcare workers or the close family who is taking care of somebody who is sick, and also during the funeral practice. And then the last risk is from survivors. So knowing this ecology and the multiple possible hosts, it's clear that future Ebola outbreaks are really inevitable. The disease itself is a very, very dramatic disease. It starts with very common symptoms, I would say, for any viral infection. So the fever, the chills, the muscle pain, the malaise, the tiredness, they can appear 7 to 14 days after exposure. And they can later have a rapid progression to a more severe disease, which is also with a hemorrhage and diarrhea and vomiting and rash. So this second phase of the disease is really extremely severe. And many people have the risk of dying here from dehydration. They can also die from the hemorrhage itself or from inflammation. The mortality of untreated Ebola virus can be very, very high. In some papers, it has been described as up to 90% mortality. The treatment with rehydration, replacement of body fluid helps reducing the mortality. But in general, it remains around 50%. And the survivors complain of sequelae. There are sequelae for the AIDS, for example, and also for the general health status. So the problem of the Ebola virus disease is for the people who are directly suffering from it due to the severity and mortality. But it's also for the society as a whole. It's a dramatic disease which is totally disrupting healthcare system and disrupting the society. So first of all, when an Ebola outbreak happens in a region which is often already a poor healthcare setting, with not a lot of healthcare workers, not a lot of hospitals, and difficult access to intensive care, a lot of the resources are diverted from the usual healthcare and are affected to Ebola. So you also see new issues happening with other health programs such as HIV, malaria, tuberculosis, trypanosomiasis. And also the primary care, the maternal and infant basic health care can be impacted. So that's really a snowball effect with Ebola. Not only you have this death and this suffering from Ebola, but also from other diseases, and also simply from giving birth. The mortality in healthcare workers is also very high because they are directly exposed to the body fluids of the sick people that they take care. So, for example, in West Africa, during the large epidemic 5 years ago, there were almost 900 healthcare workers infected. And this number has to be looked at in the context of the number of healthcare workers in these three countries in West Africa, Sierra Leone, Liberia, and Guinea, which are already countries with a limited number of healthcare workers. So for them, it was really a huge impact on the healthcare workforce. And the case fatality rate was 57% in this population. And then there has been also some calculations of the higher risk of being infected in healthcare workers. So in Sierra Leone, for healthcare workers, it was multiplied by 100, the risk of infection. And in average, if you look at war papers or reviews, it's multiplied by 20 to 30. So dramatic impact. It had also some additional impacts outside of the health, so on food insecurity, on education with many schools closed, and especially on girls, and also on many children losing one or two of their parents. The last thing that I want to show about the disease itself is the fact that the number and the frequency of Ebola outbreaks has increased in the past years in sub-Saharan Africa, and today Ebola has become a permanent threat. These outbreaks that are illustrated by the blue bars on the graph were relatively rare in the past century, but we have seen more of them in the more recent years. And not only they have been more frequent, they have also been larger. So this is really now becoming a very serious risk for public health in sub-Saharan Africa. So since 1976, there has been around 30 outbreaks. And the cost is also very high for these countries. The economic cost is huge. The calculations that were made about the cost of the Ebola outbreak in West Africa was almost 3 billion direct cost and 53 billion direct and indirect costs because the economy was totally disrupted. In 2021, we have already seen 2 outbreaks. So it was small outbreaks, nevertheless, 2 already in the same year. So a small outbreak in Guinea with so far 18 reported cases, and there has not been a recent reported cases. So there is a countdown period which is ongoing before WHO is able to declare this outbreak over. After the last case, there is always this countdown period of 42 days before being able to say it's finished. And there has been an outbreak of a similar size in DRC, in North Kivu, with 12 reported cases, and this one was declared terminated on the 3rd of May. Even if these outbreaks were small, here you see again that half of the people unfortunately died from the disease. Fortunately, there are now opportunities for the prevention of Ebola. And there are Ebola vaccination strategies that can be implemented. And I will first cover this slide, which is really about Ebola vaccination strategy in general, and then I will focus on the vaccine that I know the best because it's the one which was developed by the company I work for. So first of all, the overall strategy. There are two complementary tools for a very comprehensive public health strategy to either contain or prevent Ebola outbreaks. And there is a distinction between what you do when an outbreak is ongoing, it has started, you have to react, and what you can do in the absence of outbreak to help preventing next outbreak or to mitigate next outbreak. So let's look first at the outbreak response. It's sometimes what we call the reactive use of vaccination. And in this outbreak response, there is a first strategy that has been evaluated and that has proven very efficient. It is the use of the Merck vaccine, RVSV-ZIBOV GP, in a ring vaccination strategy in affected areas. In a ring vaccination strategy, you have to identify a person who is sick, to identify the contacts and the contacts of contacts, and you vaccinate people like this. It is what is called ring vaccination strategy, generally complemented also by healthcare worker or frontline worker vaccination. What WHO has proposed is to complement this ring vaccination strategy with the Merck vaccine in the hotspot by a complementary strategy with the Janssen vaccine regimen around this ring. And it has been defined that people who are living in affected areas or neighboring areas, but who are not eligible for the Merck vaccine, should be vaccinated with the Janssen vaccine. The idea is to make a second barrier to avoid the spread of this outbreak. Then there is another strategy for real prevention in the absence of outbreak. So for the Merck vaccine, it has been proposed to use the vaccine doses that are available and close to expiry in highest risk zone. And for the Janssen vaccine regimen, it has been proposed to vaccinate the national response teams, the international responders, the lab workers with possible exposure, and also people working in Ebola treatment research units. So this is already a good number of people who are involved in healthcare. A large preventive vaccination in the absence of an outbreak has not been recommended. These are very recent recommendations that were first proposed in 2019 by the SAGE working group, by the SAGE Strategic Advisory Group of Experts of WHO in 2019, and that were confirmed more recently in March 2021. So based on that, I will show you a little bit more about our vaccine. So this is our vaccine. It's a monovalent vaccine against Ebola Zaire, and we call it a vaccine regimen because it comes with 2 doses of vaccine, which are 2 different vaccines. The first dose is called adeno-26-ZIBOV or Zabdino, and the second dose is called MVA-BN-PHILO or MVA-BA. These 2 doses have to be given intramuscularly, and they are based on non-replicating viral vector vaccines. These vaccines are both based on platform technologies. So the adeno-26-ZIBOV is based on ADVAC technology from Janssen and is encoding for a glycoprotein Mayinga. The MVA-BN-PHILO is based on Bavarian Nordic's MVA technology, and it's encoding for several glycoproteins, EBOV, Sudan, Marburg, and Staphylovirus. Nevertheless, the overall regimen is only indicated for Ebola Zaire, and it's indicated in anybody from 1 year of age. I said that these 2 vectors are non-replicating, and this is a useful information because it's favorable in terms of tolerability and safety profile, and it also allows use in some vulnerable populations. The development of our Janssen Ebola vaccine is a long story already because it started really 6 years ago with a lot of events. And during my presentation, I will explain you this to further details. But let's first look at the start. How does all this start? In Janssen, we were having some research, some early research on filovirus vaccines in general. And when the Ebola outbreak was declared in 2014, we decided to accelerate the development. So for this acceleration, we decided that it was simpler and faster to focus on a monovalent Ebola vaccine. So instead of continuing to develop a multivalent filovirus vaccine, we really focused on monovalent Ebola Zaire vaccine. And we received funding from NIH, from IMI, which is Innovative Medicine Initiative, an initiative from the European Commission, and also from BARDA in the US. And this external funding has helped complementing our own internal funding and has really helped developing this vaccine regimen. We have based the development of this vaccine already on a very large clinical development program. Since 2014, we have had 23 studies, 23 clinical studies that were either sponsored by Janssen or sponsored by our partners, phase 1, 2, 3. 8 of these studies are fully completed, and these studies were done in Europe, in US, and in Africa, with the majority of them in Africa. 14 countries were involved: Kenya, Uganda, Tanzania, Côte d'Ivoire, Burkina Faso, Mozambique, Nigeria, Guinea, Liberia, Sierra Leone, Mali, Senegal, DRC, and Rwanda. And we are really proud of having been able to conduct the vast majority of the development of this vaccine in the countries that need this vaccine. There is also a diversity of participants who have been included in the studies. We had adults, but we also had older adults up to 70 years of age. We had HIV positive adults, children down to 1 year of age, and now we have an ongoing study in infants 4 to 11 months. We have also another study which is currently ongoing in pregnant women in Rwanda. It has started in 2020. overall, with this large clinical program plus additional projects, we have a cumulative experience of more than 200,000 vaccinations with this Ebola vaccine regimen. So a very substantial experience now. In DRC, at the time of the epidemic that started in 2018, we set up a large-scale study with 20,000 participants. More or less at the same time, we also started the vaccination campaign in Rwanda. And more recently, we have started a new large early access trial, which is sponsored by WHO in West Africa in response to the recent epidemic in Guinea. And this one has just started, but the objective is again to have 200,000 more people vaccinated in up to 6 countries. This is the tolerability and safety profile of this vaccine regimen. So you have two tables on the slide because we have evaluated reactogenicity and safety after the first dose of the regimen, adeno 26 above, and after the second dose, MVABN philo. So the adverse reactions to this vaccine are very common adverse reactions commonly observed with vaccines. Most of them are local adverse reactions such as pain, redness, pruritus, and swelling at injection site. And the general adverse reactions are also common like fatigue, headache. We have not seen anything qualifying as a signal, only common reactogenicity. This is the same thing, but for children. And in children, the safety profile of both vaccines has been very similar. We have seen a little bit more fever in children than in adults, and with an increasing frequency of fever when going down into the age. Nevertheless, still rare and really aligned with what you see with other common childhood vaccines like meningo vaccination or recently have also observed rare febrile seizures in children under the age of 7 years. But this observation was rare, so it was done in the post-approval experience only when we vaccinated massive numbers. Now, let's look at the immune response to this vaccine regimen. So the vaccine regimen has been evaluated with different intervals between Zabdino and Mviabia. But the main regimen is the regimen with 56 days, meaning 8 weeks of interval between both doses, because it's the most favorable one. Here in this table, you see the results from several studies which were conducted in different parts of the world and which are either phase 2 or phase 3 studies. The peak of the immune response is measured 3 weeks after the dose 2. And you see here that the level of the humoral response measured by antibodies, binding antibodies against the glycoprotein dosed by ELISA was very high, between 3,800 units to 10,100 units, and even 11,000 units. In children, we also have measured this humoral response with binding antibodies in different studies, which were all in Africa. And the responses in children were to the same level than the responses in adults, or even higher, with rates from 9,000 to 22,000. So overall, what is really interesting is that the response rate is very high. 98 to 100% of people respond to the vaccine across very diverse groups. The GMCs are higher in children, And what we have seen is that also these response rates are maintained at least for 2 years in adults. Now I will pause to share with you a short video.
Mistrust or armed conflict. In such circumstances, where the demonstration of vaccine efficacy in a human clinical trial is technically, logistically, or ethically not feasible, the US regulatory agency FDA allows that an immunological marker that correlates with protection from disease in a suitable animal model can be used to infer similar protection in humans. This is known as the FDA Animal Rule. The principle was also adopted by similar regulatory guidelines, including the European Medicines Agency's approval under exceptional circumstances. Janssen has developed a 2-dose vaccination against Ebola virus disease. The regimen consists of 1 dose of Zabdeno, followed by a dose of Mvabia. As an innovative alternative to a large clinical efficacy study in humans, Janssen has determined the protective effect of its vaccine candidate by comparing the immune response in humans to the immune response required for protection in a non-human primate model. For Ebola, a fully lethal Ebola virus challenge model in cynomolgus monkeys is the most relevant animal model, with similar Ebola virus disease symptoms as in humans, but with even more aggressive disease course with higher mortality. In that way, the model may even underestimate the potential effect of the vaccine in humans. Let's take a closer look at how the animal model was set up. A group of animals received the Janssen candidate Ebola vaccine regimen, while another group served as non-vaccinated controls. All animals were subsequently infected with the Ebola virus. The serum level of binding antibodies, as well as the survival of the animals in both groups, were evaluated. Binding antibodies induced by the vaccine regimen against the Ebola virus surface glycoprotein were identified as an immunological parameter correlating strongly with protection. At the clinical vaccine dose, 100% of the vaccinated animals were protected. Non-vaccinated animals did not raise antibodies and succumbed. To further examine the relationship between immune response and protection, lower doses of the vaccine were tested to draw a curve relating the level of antibodies to the survival rate. The relationship between the immune response and the survival probability in the non-human primates was assessed by logistic regression analysis. Vaccination in humans induced similarly strong anti-Ebola virus surface glycoprotein binding antibodies. By comparing the vaccine-induced antibody levels in humans with the antibody levels in the animals, A method called immunobridging protection in humans could be inferred based on the observed relationship between antibody levels and the survival rate in the animals. The width of the confidence interval is a statistical expression of the uncertainty around the observation. To ensure that the observed result was sufficiently robust, it was agreed with the US and EU regulatory agencies that the lower limit of the 95% confidence interval should be at least 20%. The mean predicted survival probability in humans based on this model was found to be 53.4%, with the lower limit of the confidence interval at 36.7%. So the preset regulatory criteria were met. The robustness of these results was further confirmed by additional sensitivity analyses, repeating the calculations in multiple subpopulations in various geographies. Based on this immunobridging, the Janssen vaccine regimen is likely to protect humans against clinical Ebola virus disease. As the exact level of protection provided by the vaccine in humans can only be established in field efficacy trials, Janssen has committed to collect clinical efficacy information whenever feasible. The Janssen 2-dose vaccine regimen Zabdeno-Mvabia against Ebola virus disease was approved in Europe in July 2020, following demonstration of its quality, safety, and efficacy based on this innovative and robust immunobridging approach. Thanks to this immunobridging, Janssen was able to make the vaccine regimen available as an additional tool in the relentless fight against the Ebola virus.
Now that we have seen all together this video about the principle of immunobridging and the basis for the approval of this vaccine, I would like to show you the long-term persistence of the immune response to this vaccine. The adeno-26-ZIBOV MVABN philo induces very durable EBOV glycoprotein-specific antibody responses measured by ELISA. We have observed directly these immune responses in some clinical trials, and we know that they last at least for 2 years. We have also conducted some modeling showing that they persist up to 5 years. It is what is illustrated in the right side of my slide on the graph. And you see the measured immune responses with the blue dots and the extrapolation with the nice curve in gray. This slide is showing the anamnestic response to adeno-26 ZIBOV booster vaccination in adults. It's based on our large clinical trial in Sierra Leone, EBL 3001. What we show here is that there is a strong anamnestic antibody response to a single dose of adeno-26-ZIBOV. It is an interesting finding because it is demonstrating that the initial vaccine regimen is inducing humoral memory. So after people have received adeno-26 MVABA 56 days apart, they were boosted with only 1 dose of adeno-26-ZIBOV, and they were boosted 2 years after the initial regimen. In less than 1 week, they displayed a strong anamnestic response with a 40-fold increase of the antibodies. Looking at the antibodies after the booster dose, we see here they persisted at a high level, even higher than the initial higher level of antibody, which was protective in non-human primate. So a very interesting finding showing that there is an opportunity to give a booster dose to people who were earlier vaccinated with this vaccine. Now let's look at something, it's the storage and the handling of this vaccine. It's compatible with the existing cold chains in Africa. We use the lower temperature, minus 85 to minus 55, so around minus 60, only for the long-term storage, so only in the large warehouses in Janssen and potentially in WHO. For the shipment, send the vaccine to Africa, or for the in storage at the central warehouse, the vaccine is kept at -20 to -15, and it is transported to the vaccination site at this cold temperature. But then, at the vaccination, vaccine can be kept for some time at 2 to 8, and this is convenient for the end user. So it's 8 months at 2 to 8 for Zabdino, it's 1 month at 2 to 8 for MVAB and Philo. Now, let's look at what's on our plate, and we still have a good number of studies which are ongoing and that we have to finish. So we have a large project called PREVAC, which is in West Africa. We have our study in infants 4 to 11 months of age, which is fully fully implemented. So all people have been included. The follow-up is ongoing. We have 2 studies in frontline workers that will be also useful because it's one of the populations with the highest exposure to Ebola. We are looking at a booster, and I have shown you the booster dose given 2 years after initial vaccine regimen. And we have also a study to evaluate the safety and tolerability of early booster. We have some long-term follow-up of our previous studies. We have done a large study with 20,000 participants in DRC, starting at the time of the Ebola outbreak. And we were having the objective of collecting safety data, but also clinical effectiveness. And this was finally not possible because the Ebola outbreak did not spread to the place where this study was conducted. We have our pregnant woman study in Rwanda, a large vaccination campaign also in Rwanda on 200,000 people. And we just started an additional study in DRC in response to the outbreak in Guinea. And it may also include up to 200,000 participants. To help with the implementation of these large programs and also with some large studies, we have used different technologies, and the participants in the studies and also the investigators really welcomed these technologies and were happy with the support provided by technologies. We have used a lot of mobile phone messaging to remind people to come for the second dose. And we have also used iris scanning for participant identification, which revealed useful, especially in countries where people do not always have an ID card. One of the settings where we have really evaluated the value of these technologies is the large deployment in Rwanda, which is called Umurinzi. And we had a high compliance, high return for the second dose of 94%. What is the regulatory status of this vaccine? So based on this clinical development program, we have obtained a marketing authorization in Europe, and it was a marketing authorization under exceptional circumstances, and exceptional referring to the data package, but this was not coming with any constraint on deployment modalities. If you look at the EMA website, you can see the European Public Assessment Report, which is really the Bible for us. And it is concluding that the overall benefit risk of this vaccine regimen is positive, that it is responding to a high unmet need for practical vaccine. And we have also the commitment to conduct effectiveness study as a post-approval commitment if the opportunity arises, because this will confirm the findings from the clinical development. We have also recently obtained the pre-qualification by the WHO for this vaccine. And we hope that based on this pre-qualification, we can now move forward to more approvals in Africa. Our hope would be to have approval in approximately 20 countries. And this would complement what we already have, because we have already this conditional approval in Rwanda and the temporary use approval in DRC. So progressively, we are in a better position for the implementation of this preventive vaccination. So as a summary, I would remind everybody that Zabdino Mviabia is a vaccine regimen indicated for the active immunization to prevent the disease caused by Ebola virus Zaire in individuals more than 1 year of age in Europe, and also pre-qualified by WHO. It has a favorable tolerability and safety profile, allowing use in vulnerable populations that are including children and HIV positive people. It has a proven non-replicating viral vector platform, and the studies in pregnant women and infants are ongoing. There is a demonstrated durability of the immune response, at least for 2 years, and even a modeling up to 5 years. The use of the vaccine is supported by the WHO SAGE recommendation as a complementary tool in response to outbreaks for the people who are not in the hotspot, so no eligible to the ring vaccination with Merck vaccine, and also for the preventive vaccination in the absence of outbreaks for some targeted people who are really at high exposure, like the national response teams or the people working in Ebola treatment centers. And we are really committed to collecting field effectiveness data upon implementation of this vaccination program in Africa. My last comment will be to show you what Bill Gates said to the world in 2015. He was really visionary because he said that the Ebola epidemic can serve as an early warning wake-up call to get ready. If we start now, we can be ready for the next epidemic. And the recent story with COVID-19 has shown that we were not yet fully ready to respond well to epidemics in the world. Nevertheless, we have made tremendous progress in accelerating vaccine development. So there are some learnings from Ebola vaccine development that have been helpful for COVID-19. Thank you to everybody for your attention. Before I really close my presentation, I would like to acknowledge all the partners that have helped us developing this vaccine regimen. So the investigators, all the study participants, my colleagues in Janssen, and also a very large number of partners who have shared with us their in-depth knowledge of Ebola, of conducting studies, and also some funding partners like BARDA, IMI, CEPI, NIH. So goodbye, afternoon to everybody.